Hello everyone. My name is Nian Liu. Thanks for giving me this opportunity to present. Line 1 is also called L1. It constitutes 17% of the human genome. Line 1 is active visual transposon that is still mobilized in the human genome. The full-length line 1 is about 6 kb long. It has 5' UTR and encodes 2 protein of 1 of 2. The full-length line 1 DNA is first transcribed into line 1 RNA, then the line 1 RNA can be translated into of 1 of 2 proteins. Then the of 1 of 2 proteins can bind the line 1 RNA and reverse transcribe into line 1 cDNA and integrate it into the genome. So through this process, line 1 has amplified itself to occupy 17% of the human genome. Currently, line 1 is still actively mobilizing in human genome. But line 1 activity is normally low in healthy cells. Line 1 activities are normally high or active in some disease. For example, in autoimmune disease, cancer, chronic viral infection, neurological disorders, and aging, line 1 activities are particularly high. Interestingly, line 1 can be also active during development. For example, in mammalian embryonic development, line 1 is active at the 2-cell stage in mice embryonic development. And when people use genome editing methods to disrupt line 1 expression, they find that embryonic development can be arrested. That means line 1 expression plays indispensable important roles. Interestingly, line 1 is also active in brain neurons. But what are the functions of line 1 is an important question in the field. This talk will address the role or function of line 1 in chromatin structure, which is also regulated by the protein SAFB. We know that the human DNA is about 2 meters long in human cells, but it is folded in 10 micrometer nucleus, which is also filled with billions of proteins, RNA molecules, and RNA binding proteins. So how is the genome folded? Whether and how the RNA or RNA binding protein influence the genome folding, and how the genome folding affect gene expression. These are very important questions in the field. SAFB protein is one architectural protein, RNA binding protein that has been identified in 1990s. SAFB was identified to be related with nuclear matrix that has been known to influence structure. But where does SAFB bind the genome and how does SAFB affect chromatin structure are not clear. Previously, we identified SAFB as one gene that can repress line 1 expression through one genetic screen. So this result make us to speculate that SAFB may affect chromatin structure through interacting with line 1 elements. To test this hypothesis, we first performed SAFB ChIP-seq, and we identified about 7,000 line 1 elements that are bound by SAFB. And we found that the SAFB bound line 1s are actively transcribed, and SAFB knockout further increase the SAFB expression. Here are 2 examples. The green box indicates the line 1 elements. You can see that through the RNA-seq, this line 1 element is transcribed with the nearby exons to form the line 1 chimeric transcripts or line 1 chimeric RNAs or the line 1 containing furin RNAs. And SAFB binds this line 1 element, and SAFB knockout further increase the line 1 elements expression. And through the UV RIP-seq, we found that cell B binds the line 1 RNAs and that are associated with chromatin. And if we use flavor pyridol to inhibit transcription, we find that cell B does not bind the line 1 DNA anymore. That means that cell B binding at line 1 depend on the line 1 transcription or line 1 RNAs. So based on the above data, we get this model. When silent line 1 is silenced, cell B does not bind line 1. When cell B is transcriptionally active, when line 1 is transcriptionally active, cell B can bind the line 1 RNA to interact with the line 1 DNA. And when cell B is knocked out, line 1 transcription gets further increased. So in other words, cell B selectively represses transcriptionally active line 1s. Next, we want to explore the role of cell B and line 1 in chromatin structure. We used the C-related technologies, especially the high C. In high C experiments, the cells are first cross-linked with formaldehyde to freeze the chromatin structure. Then restriction enzymes are used to digest the genomic DNA into pieces, but the DNA that are close to each other will be filled and marked with biotin and ligated together. So only DNA that are close to each other can be ligated. Then we isolate this junction DNA or the ligated DNA fragments. We can, through sequencing, we can tell which DNA is close to which DNA. So that will give us the three-dimensional genome structure. So the high C normally identifies compartment, TED, and loop at different resolution. TED is also called topologically associating domain. That is the chromatin structure at the 100 kb to 1 MB resolution. TED is the basic unit of the chromatin structure that influences transcription. So we performed high C in control and sub knockout cells, and through high C we can identify more TED numbers when cell B is knocked out. Interestingly, the line 1 elements are enriched at the strengthened or increased TED borders. Here is one example. Here is the cell B bound line 1 at the TED border, and the cell B knockout further strengthens this TED structure. And such cases are very pervasive or widespread. In total, we identify over thousands of cell B bound line 1 that forms the TED structure. And cell B knockout further strengthens or increases the strength of the TED boundaries. This means that the line 1 associated TED boundaries are widespread. Interestingly, we find that PL2, RNA polymerase 2, are enriched or broadly distributed over the line 1 elements. And cell B knockout further increases the PL2 occupancy. That means cell B inhibit PL2 accumulation or enrichments over line 1s. In comparison, the canonical architectural proteins such as CTCF or cohesin component RAD21 are not enriched over the TED borders. That means the RNA polymerase probably contributes a lot to the TED boundary formation. We also performed acute inhibition of the transcription by flavor pyridol treatment, and we found that when the transcription was inhibited, the line 1 TED boundaries got impaired. That means the line 1 boundaries are dependent on active transcription or PL2 occupancy. And we also performed Cas9 to delete the line 1 element, and performed the high C and found that the line 1 deletion can severely impair the TED structure and reduce the transcription of the genes within the TEDs. Overall, based on the above data, we got this model. The active transcribed line 1s can accumulate PL2 and express the line 1 chimeric transcripts. And cell B can sense or recognize the transcriptionally active line 1 elements to inhibit PL2 accumulation. And when cell B is depleted or knocked out, we observed more PL2 accumulated at the line 1 elements, which contribute to stronger tail structures, which will influence the nearby enhancer promoter interactions and influence the gene transcription. So our results identify a novel function of line 1 as the TED boundary elements. This process is influenced by the line 1 transcription homeostasis. In the end, I want to thank the people at the lab for doing the work. I want to thank my collaborators and thank you for your attention.